express my apologies for not being able to be present in person. I would have liked to have made my first trip to Sri Lanka to meet many of you in person. Osmi is the only one I've met in person, but I hope that this is the beginning of a longer-term interaction, and perhaps we'll see some of you in La Jolla, and perhaps others of us will be able to visit and establish collaborations that endure for more than just a brief training session. By way of a little bit of introduction, so that you know something about what I am and what I'm not. I'm a zooplankton ecologist and biological oceanographer, and my principal expertise is the population dynamics of the multicellular zooplankton. By the way, if there's ever a problem with the sound or the video, someone will need to alert me in some conspicuous way, because I have just a very small image of your auditorium on the screen, and so you'll have to let me know if there's a problem. Okay, good. So what I'm not is I'm not a fisheries biologist or ecologist. And I understand that there are many people at NARA who have interest in pelagic fisheries and demersal fisheries. That's just not in the realm of my expertise, so that's not a topic that I'm going to be covering, but there certainly is deep expertise in that area in the community at Scripps, between Scripps Oceanography, the Southwest Fisheries Science Center, which is a National Marine Fisheries Service laboratory, and other nearby locations. I am the founding principal investigator of a long-term ecological research site called the California Current Ecosystem. And alas, I'm not going to have time to tell you about that today. This program is one that is dynamic. It evolves over time as the research questions and the people involved evolve. And I would actually like to encourage my colleagues there to think about whether establishing a long-term ecological research site might be appropriate for Sri Lankan coastal waters. But that's a subject that we can discuss perhaps on another occasion. For 30 years, I served as the curator of the pelagic invertebrate collection, i.e. the zooplankton collection at Scripps. And that seems to be the largest such collection in the world. And you'll hear from me a few comments about the critical importance of sample archives for validation purposes, for voucher specimens, for unforeseen and unanticipated needs, future research needs, and the need, the imperative to maintain field collected specimens in a condition such that they can be used for a very long time to come, to be used by posterity. Another element of my research interests are concern an autonomous robotic vehicle that we've developed at Scripps. I call it the Zooglider. And this is a device that both optically and acoustically senses the zooplankton. And I'm afraid that that's not terribly germane to today's presentation, but I would be happy to talk with others, with anyone about it at any point that you might like. And I've published a few scientific papers. Okay, so what I would like to do today is to cover several topics, and these range across different aspects of biological oceanography. And I have to say at the outset that for most of these topics, this is not my primary research expertise. And in fact, the area, the topic number 5 at the bottom, the mesozooplankton, which is my primary research expertise, is the one topic that I'm going to be speaking least about. And this is just, I think, in the interests of time and priorities, and my reflections on what I think would be really fundamental to creating an appropriate infrastructure measurement observing system in Sri Lankan waters. So I'm going to go through each of these topics, and what I would like to do is encourage people to ask questions along the way, if you wish. That can be a little bit awkward in this format, but what I will do is when I reach the end of the first section, I'm going to stop, and I would very much like to have a back and forth dialogue at that point. So at the end of each one of these sections, let's plan to interact to the maximum extent possible. Okay, so there's the game plan for this morning for you, this evening for me, and let's forge on. I think it will probably be appropriate to take a break as well, perhaps a physiology break, and I'll let Lahini be the mediator and decision maker on when the right time for that is. Okay, so let's begin with the phytoplankton. And this is an image, a satellite remote sensing image of inferred chlorophyll A concentrations in the eastern sector of the Pacific Ocean, the eastern part of the California current system. And the color shading here is logarithmic. It's chlorophyll inferred from absorbance ratios, generally a blue-green ratio, modified in different algorithms. But the important point is that there are very strong gradients in the cross shore. I don't know if you can see my cursor. Can you see the cursor here?
Yes.
Was that a yes or a no? That's a yes. I see the thumb. Thank you. So we see very elevated concentrations close to the coast, which at this time of year is largely due to coastal upwelling, the coastal upwelling process. And we see strong cross-shore gradients where some of this newly upwelled water elevated in chlorophyll A is being advected into the offshore region. And note that there are very sharp gradients here and very strong differences. The magenta color is about 2 orders of magnitude lower in chlorophyll concentration than the orange shade that you see here. So we see strong spatial gradients. We see a gradient from north to south with considerably lower concentrations of chlorophyll in the southeastern part of this domain. By the way, San Diego is right here, and Scripps, where I'm located, is right about where this arrowhead is at this moment. So there are regional gradients in the alongshore, in the cross-shore dimension, and of course there are rich features here, mesoscale and submesoscale features. Some of them include counterclockwise eddy here, another counterclockwise eddy here, perhaps an eddy dipole down here. Okay, so my point is that using satellite remote sensing and inferring chlorophyll concentrations from space, we can extract a lot of very useful information. And I know that there is a strong remote sensing group at NARA. Why is it that we care about chlorophyll A? And the chlorophyll itself is not so interesting, but chlorophyll as a proxy for the biomass of phytoplankton becomes much more interesting. And that's because, for one, the rate of primary production is often proportional to chlorophyll A biomass. By the way, when I use the Greek symbol alpha, I mean proportional to. And I'd say is often, but not always, but in many cases, the spatial variability of chlorophyll A has some indication of the spatial variability in primary production rates. Also, elemental export of carbon, nitrogen, and other biogenic elements, bioreactive elements, can be proportional to chlorophyll A biomass. Another reason why chlorophyll concentrations are useful. The structure of food webs, including in some cases the successful first feeding of larval fishes, may be proportional to chlorophyll A biomass. There are many assumptions within that statement, but it stands as essentially true. And chlorophyll concentrations can also be indicative of eutrophication. So this is it's a basal fundamental property of pelagic marine ecosystems, as most of you will certainly know, and it can be sensed remotely. And so one question might be, why should we not rely on satellite remote sensing alone for this important basal measurement near the base of the pelagic food web? Well, there's some issues with remote sensing, and one is that it always requires sea truth measurements to calibrate the algorithms used to infer phytoplankton chlorophyll, biomass, primary production rates, carbon export, etc. The sea truth in situ measurements are vitally important, and they're often regionally specific. The satellite sensors sense only the near surface phytoplankton concentrations, and in many parts of the ocean, especially would think the stratified Bay of Bengal and elsewhere in some of Sri Lanka's waters, the deep chlorophyll maximum will be a strongly expressed feature that would largely be outside the reach of the satellite sensor. These sensors do not resolve phytoplankton community structure. And there are some multispectral methods that are starting to be used, and there's a lot of algorithmic development, but at this point, there's no mature, reliable means to assess not just the concentration, but the composition of the phytoplankton assemblage. And then, of course, this approach also suffers a disadvantage that we can only use the images on nice cloud-free days, and in daytime is essential because we're measuring the reflectance of selected wavelengths off the ocean surface. And there are many parts of the ocean where this cloud-free daytime imagery presents real challenges. So this is what leads me to focus on direct measurement of chlorophyll A concentrations using water samples. And for that purpose, I would like to just remind people of a few fundamental properties of chlorophyll A. Remember that there are many chlorophylls, but chlorophyll A is ubiquitous as the primary photosynthetic pigment that's used by all phytoplankton groups. Now, most phytoplankton and other algae also have accessory pigments, but they all have chlorophyll A. So chlorophyll A can be used as a proxy for the biomass of phytoplankton. However, there are a number of assumptions involved, and that making the conversions from chlorophyll to biomass requires knowledge of the conversion of chlorophyll to a unit that might be of greater interest to you, either the organic carbon content or the nitrogen content, or perhaps some other element. And these ratios, the carbon to chlorophyll ratio, the nitrogen to chlorophyll ratio, et cetera, can be highly variable. So it requires some attention. Chlorophyll A is measurable at very low concentrations when using the technique of fluorometry. Now, chlorophyll A and other chlorophylls can also be measured by other methods, including spectrophotometrically, by HPLC, and by remote sensing, as we've just seen. But the fluorometric assay for chlorophyll is generally the most sensitive approach that we have currently. Now, there's a rather important difference. Sorry, was there a question?
No, Mark. No.
Nope. Okay. So there's an important distinction between extracted chlorophyll A and in vivo chlorophyll A. And what do I mean? Well, by extracted chlorophyll A, I mean a situation where you take a water sample, and since we're focusing on the plankton here and not Stewart's benthic macroalgae, you take a water sample, generally filter that sample, extract the chlorophyll on the phytoplankton that are retained on the filter in organic solvents, and then measure the concentration of chlorophyll in solution. That's extracting the pigments from the cells. That's very different from measuring the in vivo fluorescence, where you take an assemblage of living phytoplankton and excite it with the appropriate wavelength of light and measure the fluorescence at that wavelength, at an appropriate wavelength of light as these cells drift by. Now, Uwe and I have moorings in the California current system where we deploy a number of fluorometers for measuring phytoplankton chlorophyll A, but that's measuring the in vivo fluorescence of the living cells. And there's some yet further complications in interpreting and correcting those fluorescence values to biomass. And I'm not going to go into that today. I'm going to focus on this first point, the extracted chlorophyll. And I'd also like to point out that there is a fairly simple and straightforward approach to easily characterizing the size structure of the phytoplankton community using a fluorometric assay, and that's size fractionating the natural assemblage. And I'll say a little bit more about that. Okay, so we've seen the importance of assessing chlorophyll as a basal metric in ocean food webs, and we see that it's a universal tag, universal marker that can be found in all of the photosynthetic groups of interest to us. Now, let's look a little more closely at how it is that we're able to utilize a fluorometric assay for determining the presence at very low concentrations. So this is the molecular structure of chlorophyll A, which I will let Lahini delve into the details of this, which are not really my expertise or my interest. But the main point I want to make is that the chlorophyll A molecule has two key structures. One is a long phytol chain, a hydrocarbon chain over here, and the other one, a porphyrin ring that's highlighted in pink over here. Okay, so the important characteristic of this porphyrin ring is that it's fluorescent if excited with the right wavelength of light. And you might note that at the center of this porphyrin ring, there's a magnesium atom, and that will become important in just a moment. Okay, so we have this wonderful porphyrin ring that has fluorescent properties, and what's the shape of that fluorescent spectrum? And here is this is the fluorescence of pure chlorophyll A extracted in acetone, 90% acetone. Remember, I said we take phytoplankton, filter them, extract the chlorophyll pigments in an organic solvent, and then measure the fluorescence in that solvent. In this case, the solution of phytoplankton chlorophyll is excited with blue light centered at about 440 nanometers. And we're measuring the fluorescence response, the emission spectrum. And you can see the fluorescence response is a function of wavelength with a very sharp peak. And the peak of the fluorescence emission is right around 667 nanometers or so, okay? It's a very, very sharp peak. And this response is considerably more sensitive than spectrophotometry. Spectrophotometry is measuring the absorption of light by the solution of chlorophyll. This is measuring the fluorescence of light. Okay, so we have a clearly defined excitation and emission peak. Excite in the blue, it fluoresces in the red. And there's the further very useful property that this fluorescence response is highly linear over a very broad range of chlorophyll concentrations. So the curve on the right is a calibration relationship that was developed in the laboratory for a particular type of fluorometer, a trilogy fluorometer, and Lahini and Uwe brought one with them. I hope it arrived intact. Did that transport go okay?
Yep. Yes,
Great.
it's here.
Okay, so this is the calibration relationship that we typically see for just that type of fluorometer, where on the x-axis is an extract of pure chlorophyll. Now, unfortunately, we have to purchase the chlorophyll from a biochemical company. We purchase ours from Sigma Aldrich and then dissolve that pure chlorophyll in acetone. We have known concentrations, and we measure the fluorescence response over a few orders of magnitude changes in concentration. And this curve does not really let you see the low concentrations, but suffice it to say that not only is the fluorescence assay useful over a wide range of concentrations, it's extremely linear over a wide range of concentrations. So this makes the assay sensitive and linear and useful for a wide range of different ocean conditions. Okay, so the fluorescence property of pure chlorophyll. Now, pure chlorophyll does not stay pure in the ocean environment. There are common breakdown products of chlorophyll, and this can happen due to grazing by zooplankton. It can occur due to bacterial oxidation. It can occur due to extreme photobleaching and other physiological stresses. So what we have here on the left is the same structure we looked at before, chlorophyll A, a phytol chain, and a porphyrin ring. But one of the common degradation products or breakdown products of pure chlorophyll A develops when the pigment is acidified. And if you watch that central magnesium atom, if that magnesium atom is replaced by a hydrogen ion, as what might typically happen in an acidic gut, we have a proton that replaces the magnesium, and we've acidified the compound. Note that the porphyrin ring remains perfectly intact. The phytol chain is intact. And therefore, the porphyrin ring, you might expect to retain its fluorescence. And in fact, it does fluoresce. But this particular degradation product, where the magnesium atom is replaced by a hydrogen atom, is called phaeophytin A. Now, there's another common breakdown product pathway where the phytol chain is cleaved off. Note that the hydrogen has replaced the magnesium, and so we have that, but the porphyrin ring remains intact. But in this case as well, this compound, which is now known as phaeophorbide A, it retains the porphyrin ring and the fluorescence capability. So you'll often see the term phaeopigments, where phaeopigments refers to phaeophytin and phaeophorbide that can arise due to these different degradation processes that I referred to. Okay, now the reason we need to be aware of this is if we go back to the fluorescence spectra, the fluorescence spectrum on the top is the fluorescence response of pure chlorophyll A that we already saw. And once again, they're excited in the blue, it's fluorescing in the red, centered at about 6xb7 nanometers. If we look at the fluorescence response of phaeophytin A, we've converted the chlorophyll to phaeophytin by acidifying just the central magnesium atom, the central part of the porphyrin ring. The lower panel shows the fluorescence spectrum, again in 90% acetone, and you see just a very, very slight shift, maybe 2 or 3 nanometers shift to the right. But the significant point is that the fluorescence spectra of chlorophyll A and phaeophytin A are very, very similar. And this means that we can analyze for both chlorophyll A and phaeopigments, actually phaeophytin and phaeophorbide, by measuring red fluorescence when excited with blue light. Okay, so the fluorescence assay is useful not only for the intact chlorophyll, but also for the breakdown products. All right, so how do we actually ascertain how much of the fluorescent signal is due to intact chlorophyll and how much of it is due to the degradation products, the phaopigments? Well, what we do is to form an extract of phytoplankton pigments, notably chlorophyll pigments, in 90% acetone. We measure the total fluorescence, and that's due to both chlorophyll plus phaopigments. We then acidify that sample with one normal HCl, with basically 2 drops of about 10% HCl. And what that does is it converts all of the intact chlorophyll to phaopigments. We then measure the total fluorescence again, and the difference of the fluorescence before acidification minus fluorescence after acidification permits us to separate the contribution of chlorophyll A from phaopigments to that original fluorescence signal. Okay, now there's a particular coefficient that we need to know to make that correction, but I'm not going to go into that. But so the principle is quite simple. Measure fluorescence before and after acidification and make the correction. Okay. So I know that this is supposed to be a training session, and it's not really feasible to try to line people up at a fluorometer, but I wanted to just briefly walk you through the steps that one would go through to make appropriate measurements in the ocean. And I hope that with the new fluorometer that you have, that you'll be able to make those measurements soon. So the process, of course, begins with sample collection and filtration. So typically we collect a water sample with a Niskin bottle or other water bottle, filter a sample onto a glass fiber filter. The designation is GFF, the type of filter that we sent together with the fluorometer. And the nominal pore size on these filters is about 0.7 micrometers. And then after you've done that filtration, you can freeze the filters if you are not prepared to do the assay immediately. You can put them in the freezer and come back. The next step is to extract the pigments in 90% acetone. So you place the filter in a 90% acetone solution. As an optional step, if you have a sonicator, ultrasonicator, or a tissue grinder, that's very useful to disrupt the cells. It actually disrupts cells and cell organelles and liberates the contained chlorophyll in the plastids a little bit more quickly. But it's been found that if you don't sonicate or grind, the pigments, essentially all the pigments will come out anyway, or nearly all of them, 98, 99%. So you then place the extract in the dark at a refrigerator temperature for 24 hours to extract the pigments. You then invert the sample to mix it, and then centrifuge and a clinical centrifuge to pelletize particulates, just a standard desktop clinical centrifuge. And then the last step is to analyze the pigments by fluorometry, doing what we just said. And by the way, in the lower right, this is a picture of your new Turner Designs Trilogy fluorometer. So you analyze fluorescence before acidification, add 2 drops of hydrochloric acid, analyze fluorescence after acidification, and then you calculate the chlorophyll A and phaopigment concentrations by the application of the calibration relationship. And we've, technician here calibrated your fluorometer just before sending it, so yours is ready to go and you should be able to get good quantitative measurements. Okay, so sometimes people express surprise at why we should even pay attention to phaopigments in the ocean, because rapidly growing, exponentially growing healthy phytoplankton, if you grow them in culture, usually show negligible concentrations of phaopigments. If you start a culture, a batch culture, incubate them under saturating light, appropriate nutrients and adequate CO2. They will grow exponentially, and essentially all the pigment will be as intact chlorophyll. However, if we go into the ocean, most of the time, in most vertical profiles, the intact chlorophyll A is strongly dominant and is much higher concentration than the concentrations of phytopigments, but there are some exceptions. And the figures that I'm showing you here are one of those interesting exceptions. These are vertical profiles of chlorophyll A on the left and phytopigments A on the right. This is from 0 to 100 meters. And both of the pigments are on the same concentration scale. And you see, and these are, by the way, repeated vertical profiles while using a Lagrangian technique to follow a parcel of water. And you see that in at least 2 of the profiles, the phytopigment concentrations are actually much, much higher, substantially higher by about a factor of 2 than the chlorophyll concentrations are. And this location is a fascinating part of the ocean. It's in the Eastern Tropical Pacific. It's the Costa Rica Dome area, which is a natural region of semi-permanent upwelling and has a very interesting zooplankton grazing community, which was the focus of study by Decima et al. So I wanted to just ask people, so what hypotheses might you have for why in this part of the ocean, why might there be substantially higher phytopigments than chlorophyll when in most profiles, in most regions, we see relatively low phytopigment concentrations? So where could the phytopigments be coming from? I can only hear a mumble. I think that was Lihini.
Yeah, I'm just trying to repeat the question.
Lihini, could you repeat the statements?
Can you hear it now?
I think you do.
Must be higher in that way because the input is coming up. Became levels are high. Became low that.
I'm sorry, it's a little muddled. It's a little hard for me to understand.
So I think, if I understood you correctly, you're suggesting that maybe there are high phytopigments because you have upwelling in that region. So, where do you think the who's got the phytopigments? Is it, you think that's phytoplankton? Decaying phytoplankton. So the hypothesis is that it's decayed phytoplankton at those depths, is what you're seeing.
Could you repeat? Could you say that once more, please, Lihini?
So sorry, the hypothesis is that it's decaying phytoplankton at that depth, the phytopigment profile.
That's a very good hypothesis, and we don't know for sure whether it's decaying phytoplankton because it's recently grazed material, because it's suspended marine snow that has been that is floating as neutrally buoyant and remaining in the water column with lots of breakdown products in it, or might be some bacterial lysis that has caused this to occur. What's unlikely to be the case, it's unlikely to be a photodegradation process because the phthalopigments are not high up near the surface where the light would be brightest. But this is the kind of profile that stimulates other questions and other kinds of experiments. And actually, if you're interested, I would refer you to this paper. Okay, so good. Let me just see, was there, yeah, I wanted to make one other point about chlorophyll A and its utility. And that's that it's not difficult to size fraction the chlorophyll A, the water samples used for determining chlorophyll A. Now, it's a very imperfect and some would say inadequate approximation of phytoplankton community structure because, because simply taking water sample and passing it through successive sizes of mesh or polycarbonate filters might give you information on size composition, but ignores taxonomic, genetic, physiological, and functional differences. So that's one drawback. However, it's simple and inexpensive. And I would say the optimum would be to size fractionate some chlorophyll samples, but then also to conduct the parallel analyses by taxonomic means. I think you have good taxonomists there using HPLC or other methods. But a simplified size fractionation protocol would entail filtering an aliquot and analyzing total chlorophyll A retained on a glass fiber filter, and then filtering a second aliquot and analyzing the chlorophyll A retained on a 2 micrometer polycarbonate filter. And we've sent both glass fiber and 2 micrometer polycarbonate filters to you. And then the difference between them enables you to distinguish the amount of chlorophyll that's in the fraction of about, say, 0.7 to 2 micrometers, the picoplankton, which are primarily Synechococcus and some Prochlorococcus, and to separate those from the larger phytoplankton, the greater than 2 micrometer nano and micro plankton. Okay, so let me show you an example of why this can be informative and why you might want to think about doing something like this in your coastal waters. This is coming back to the California current system that we know and love. This is a different type of satellite remote sensing image. Again, it's chlorophyll concentration, inferred chlorophyll concentration in the background from absorption ratios. And these numbers, 1, 2, 3, 4, refer to sampling localities as we followed drifters that were being advected offshore. And so note, one is in this, this highly enriched, elevated chlorophyll concentration region of about 10 micrograms chlorophyll per liter, and 4 is off in the offshore in something that's close to about 0.1 or 0.2 micrograms chlorophyll per liter. Okay, so the satellite image is permitting us to infer changes in concentration. But what's shown below is a plot showing the proportional composition by different size fractions of water samples that were taken as the ship moved from our sampling locations 1 through 4, where 1 is on the right close to the coast and 4 is on the left further offshore. And this is then a representation of the average composition of different phytoplankton size fractions to the total chlorophyll. And this is done by our colleague Rolf Gericke. He does 5 size fractions. In the previous slide, I referred to simplifying this to just 2, but he separates the phytoplankton chlorophyll into less than 1 micrometer, 1 to 3, 3 to 8, 8 to 20, and greater than 20 micrometers. And the important spatial difference here is if we start out at cycle 1, at sampling location 1, which was close to the coast in the elevated high chlorophyll concentration region, we see that it's overwhelmingly dominated by this purple hue, which is the the large phytoplankton greater than 20 micrometers. It's about 70% of the chlorophyll biomass is in the form of the largest cells. As we go into the offshore, we go off to sampling location number 4, the furthest of the offshore, the pattern flips. And if we look at this red line, below the red line is everything between glass fiber filter and 3 micrometers. So now we have the smaller cells, the less than 3 micrometer cells, now about 60% of the total chlorophyll biomass. And so we have not only the change in concentration, which we can get from space, but the shift in the size composition of the phytoplankton community that we can only get by direct measurement. And I would suggest to you that although it's always nice to have multiple size fractions like this assay could be simplified to just glass fiber filter and greater than 2, and you would still get an important separation of the picoplankton in your coastal waters from the other forms of phytoplankton. Let me show you one other example of how a simple size fractionation can be informative. And this is, again, the value of separating the picoplankton from the larger phytoplankton. The 2 graphs that you see on the left are based on measurements made by the Ocean Institute and very close to the coast. The Ocean Institute is a private entity that is an educational organization that takes school children out on vessels to introduce them to the marine environment and acquaints them with some of the fauna and flora of the water column, fauna and flora of the seafloor. Well, so they've teamed up with us to make, not quite daily, but I think a few times per week, if the sampling goes well, they will take water samples and they will filter 1 fraction through a glass fiber filter. That's what we call total chlorophyll A. It's approximately total chlorophyll A. And then another aliquot is filtered through a 3 micrometer filter. And the upper panel is what I call an El Nino neutral year, where, and this, by the way, is calendar months, January through December. The data stopped at the end of September. But you see that the blue line is the total chlorophyll, and we see a lot of variability. The orange line is the chlorophyll that's greater than 3 micrometers. So in this case, it's greater than 3, not less than. Greater than 3 micrometers. And the striking point is that when there are big pulses of phytoplankton, the big blue pulses, for most of them, they're explained by the orange peak, by the greater than 3 micrometer cells. And then over here, this is, we're out here in July and August. Now we see much more difference between the orange line and the blue line, which means that a larger fraction of the phytoplankton is the less than 3 micrometers. So in this normal non-El Nino year, most of the time blooms, episodic blooms that are frequently associated with coastal upwelling filaments, that those blooms are dominated by large macroscopic phytoplankton. And they have, there are very different grazers that utilize those cells. Now, if we look at the lower panel, this is, again, an annual measurement for a different year. The first part of this year was El Nino, 2015 into about May of 2016 was a lingering El Nino, a moderate strength El Nino. And the first point you'll notice is that if you look at the height of the blue curves relative to the height of the blue curves in the El Nino neutral year, that the peak chlorophyll concentrations, total chlorophyll concentrations, were substantially lower than in the neutral year. So we know that typically upwelling either relaxes, or if the winds are upwelling favorable, the nitrocline is suppressed deep enough that the new nutrients supplied during those wind events are reduced. But the net effect is that there's a lower nutrient supply and lower phytoplankton biomass. That's the first point, is lower concentrations, which we can see just from the blue line. But now let's go back and compare the orange line with the blue line, and you see that throughout this El Nino period, most of the phytoplankton, these weren't real blooms, but they were small increases in phytoplankton concentration, that those were not tracked by the greater than 3 micrometer cells. In other words, most of the phytoplankton that was proliferating to a modest extent during the El Nino conditions were smaller cells, were the picoplankton, not the larger cells that proliferate during the upwelling blooms that we saw in the previous year. And so these can be very important, useful characteristics that help us characterize some basic properties of the food web, where the picoplankton tend to be especially important for the microbial food web in the water column and for recycling processes. And the microphytoplankton, the larger cells, are often more important for the metazoan, the multicellular animal food web, for carbon export, and sometimes more significant for fisheries. So this is why I think that a simple size fractionation of your regular chlorophyll A measurements by fluorometry could be very informative in Sri Lankan coastal waters. Okay, so that's module 1. By the way, that's the longest one. These aren't all going to take that long. But I'd like to stop and see if there are any questions or any points of clarification. If you'd like me to go back and show and explain a slide, feel free to ask.
Anybody has questions? Mark, I was wondering how whether you could quickly connect it to, I mean, you say sometimes fisheries, but could you quickly connect like size fractionated chlorophyll to sort of the planktivorous fish populations, or is that not a really clear connection?
Well, it's a contingent connection. So the connection is that in these waters, the small planktivorous fishes, that's primarily sardines, Pacific sardine, northern anchovy, and jack mackerel, that they are especially dependent upon small microplankton cells in their first feeding stages. Excuse me. So after they emerge from a yolk sac stage, where the yolk is their primary source of nutrition, there is the critical first feeding stage where they have to find the right type and the right size of microplankton. It could be either phytoplankton or sometimes microzooplankton in the right concentrations. And if they don't find them in a short period of time, they starve and die. And presumably this has consequences for recruitment success. So the size fractionation method would enable someone to say, yes, the phytoplankton was dominated by larger cells or was dominated by smaller cells. It would not definitively say that these were the right type of cells that the first feeding larvae must acquire, but it would be an indication in that direction, and it would certainly be much more informative than just the bulk chlorophyll without the size information.
Right, so you could potentially at least infer that the ocean conditions were good for either microphytoplankton or not, potentially, right? Even though these might not be the right microphytoplankton.
That's correct. And another way to approach this would be as adaptive sampling. So if the size fractionated chlorophyll measurements were made on a regular basis, and there was a sudden change where there's a pulse of much larger phytoplankton cells, then at the next sampling location to go out and take additional samples that might be analyzed by microscopy or might be analyzed by electronic particle counter or another means to focus on, or, flow cytometry, imaging flow cytometer, whatever device might be available. But to use this as an indicator, as a sentinel of a change in the ocean that would then lead you to go out, take additional samples to further probe the change in the food web structure.
Okay. Thank you, Mark. Yes, so I think one of the key take homes anyway for me is that, this kind of analysis gives you a good indication of what's happening at the base of the food web and potentially in terms of nutrient supply, which then has consequences for the higher trophic level, right?
Right.
Questions? So I think many of you measure more with
Hi, Mark. Nice to see you.
Oh, hello. Hi, Osmi.
Very nice. I think your lectures are really useful. I'm not an expert, but I'm sure all these people working on chlorophyll and phytoplanktons, they are getting a lot of information from you. And if you have any books on this, I'm sure you might be having, just send us the link we can get for the NARA library and for your method. So I have a few questions. One is there's ocean acidification going on in many places, but not in Sri Lanka. From our data, there is hardly any pH increase noted for many years now. But this pheophytin change or the degradation of chlorophyll to pheophytin, does it happen where there is lowering of pH in the ocean? That is one question. And the next one is we all the nitrogen and phosphorus concentration are the main the indicators of whether there is higher plankton or lower plankton density. So we have from land-based sources, we have the agricultural waste coming in, the fertilizers, and also the domestic waste coming through our falls and the rivers and into the estuary. So have you done studies to identify, which is more, responsible, agricultural waste or the domestic waste? And what are the other thing is upwelling, the upwelling. But from land-based sources, what is the, the indicative or factors that influence phytoplankton production or the activity on the ocean?
This is very frustrating because I think I understood about every third word. I think, first of all, regarding books and references, I will provide a list of citations, full references to everything that I've referred to here, and add some that I think might be informative. I think your first question had to do with ocean acidification, and I couldn't really understand. It's just, the problem is the sound is breaking up, and so I don't get full sentences. And did the second question have to do with phosphorus?
Yeah, I can ask again. Hello, you can hear me now?
I can hear you,
Yeah,
yes.
Yeah, you were telling about it, magnesium being replaced by hydrogen.
Oh, okay, yes.
Yes, and I don't know, maybe there is more chemistry to it. Maybe magnesium has is double valency, whereas hydrogen has single. But is the hydrogen change of hydrogen due to acidification or some other factors? If it is occurring in the natural environment.
To my knowledge, the changes in ocean pH are nowhere close to what would be needed to acidify a chlorophyll molecule. And the chlorophyll molecules are in plastids and they're protected by membranes, so they're not exposed to the ambient pH. So we don't have to worry about ocean acidification converting chlorophyll to phthalopigments, though that's at least not in the near term.
Okay, Mark, I think the other question was whether you could differentiate the importance of different nutrient sources. So either upwelling versus agricultural nutrient delivery, or I think domestic waste related nutrients, and how that might impact the food web.
This is beyond my personal expertise, but I can say that from a distance, the situation in Southern California is that this is a very arid landscape. We don't have any major natural rivers anymore. So terrestrial nutrient discharge via riverine sources is really of very little consequence. There is, however, both diffuse discharge of sewage and runoff, and also point source discharges associated with sewage treatment plants. And the level of sewage treatment is supposed to be at least secondary treatment in these waters, which means that the nutrient loading is relatively low. So this would, I would say, Osmi, if we were very close to the coast in the first, let's say 1 to 5 kilometers, I think that especially the diffuse source runoff from land could be an important factor. But as soon as we get into the open coast strong upwelling zone, the volume of nutrients that's coming up from below via upwelling is orders of magnitude greater than the terrestrial runoff sources. In this part, and I would say very important, that's for this part of the ocean. That's highly dependent upon the local topography, rainfall, river sources in different parts of the world.
And I think, I'm sure the people here at NARA do this, but one way to get at some of that is just looking at the species of nutrients. So ammonia versus nitrate, right? But typically nitrate, in our system is primarily upwelling derived, and the ammonia is a better indicator of external.
Yes, and to look at different forms of organic nitrogen, urea, notably included among them, as well as ammonium, I would quite agree.
And if you really wanted to get complicated, what I like to do, you can use nitrogen isotopes as well as a very effective tracer of sewage agriculture versus nitrogen.
Okay, shall we move on?
Yeah, then we have a question.
Oh, sorry, was there one more question?
Yeah, we're getting now.
We're moving on. Okay. All right. So I'd like to
Wait, Mark, she's up. There's a question coming. Sorry,
Okay, sorry.
I just handed the microphone. I know you can't see.
Sorry, everyone.
Could you please speak really directly into the microphone?
She is, just one second. She's gonna come to the computer. Do you
That
mind?
works better. Thank you.
Sorry.
Hello.
Hello.
Hello. Yeah, that in the normal year, that it means that in a neutral year, you said that there is a like 20 microliters per like that huge amount of chlorophyll concentration, and in the El Nino year, there is only 0 to 5. That huge decrease of that chlorophyll. Can you please explain why is that huge decrease due to this El Nino in the graph?
I caught
Mark?
it. If
Yeah, Mark, so she's just asking why the chlorophyll concentrations. Sorry, I'm gonna plug my Mac in soon, but why the chlorophyll concentrations are different between the 2 years. So why does the El Nino year have lower chlorophyll than the neutral year?
we think that it has to do with a reduced nutrient flux from below. Weaker upwelling, either weaker winds or stronger density stratification, and therefore a greater barrier to nutrient flux from below. But in essence, we think it's nutrient limitation. Okay. Well, forgive me, I'm curious. Do you have a strong expression of El Nino in your coastal waters?
Do you see El Nino signal in Sri Lankan waters? I think you do. But what is the signal typically? Just a, how do you detect it? It's worldly. Okay, so I guess it's a very strong temperature signal, a warming signal. Which they can detect as coral bleaching events.
Yes.
Did you hear that?
Yes, I did.
Okay,
Yes.
okay, yeah.
Okay, well, might be an interesting topic for future discussion. I think I will we'll move on, and I'll have to go a little faster. I wanted to bring up the importance of measures of optical transparency of the water column, because the optical transparency is affected by a range of different properties, and that includes the concentrations of living phytoplankton, as we've just seen, concentrations of detritus and other forms of marine snow and other forms of detritus, dissolved organic matter, and then suspended inorganic matter, including dust and resuspended sediments. And the optical field, the optical transparency is something that can be measured by very complicated methods or by a relatively simple method. And some of the answers come out to be essentially the same. But that the merits of characterizing the optical transparency include the fact that decreased transparency can be indicative of eutrophication effects in certain circumstances. Transparency can be proportional to phytoplankton biomass. It's also very important for the encounter radius and the encounter volume that visually hunting predators have when they're hunting for prey. So we're thinking about fish here, sight hunting fish, and primarily plankton feeding fishes. If you have identical concentrations of zooplankton, let's say copepods, in two different water parcels, but one water parcel is optically much more opaque than the other water parcel, then the effective prey availability to the fish can be dramatically different. The concentration of prey can be the same, but the availability of the prey can be very different. And it's quite sensitive to the optical properties of the water column. And then also, we frequently need to define the depth of the euphotic zone for making calculations and measurements of various kinds. So the optical properties of the water column are something I would urge you to measure. And this is an area that is completely outside my expertise. It's a very advanced field. There are some very accomplished practitioners at Scripps and elsewhere, and there are many different types of instruments and approaches that can be used to characterize both suspended and dissolved substances, that can be used to differentiate the inherent optical properties from the apparent optical properties of the water column. And many of these optical measurements are very relevant to the calibration and validation of satellite remote sensing methods. And I think these are measurements that are worthy of attention. If you have someone with this appropriate expertise, if someone with the appropriate expertise is in the audience, my apologies, because this is something that is not my field, but suffice it to say that there is a longstanding, one of the oldest and simplest methods that we still use in CalCoffee and the LTR program, and that's the Secchi disk depth. And of course, the Secchi disk is a 30 centimeter diameter white disk. Earlier on, it was alternating black and white. And the Secchi disk depth is simply the depth at which the disk disappears when lowered into the ocean or the lake, or a lake. And the useful property is that the depth at which it disappears, and here's a 30 centimeter diameter white Secchi disk. I think it looks like it's probably just been retrieved from CalCoffee. But it's lowered over the side. One or two observers carefully watch until it disappears, record that depth. And that depth, which is Z sub SD here, the Secchi disk depth is proportional to what we really want to know, which is the beam attenuation coefficient and diffuse attenuation coefficient of the water column. What's in the numerator in this equation are constants. When we have to know the constants, but once we know them, then what is important is what's in the denominator. And so the diffuse and beam attenuation coefficients are what we require and we're interested in order to characterize this optical field for sight hunting fish, or eutrophication effects, etc. And that is proportional to the Secchi disk depth. Very simple measurement. And of course, there's a arithmetic relationship between the depth of the euphotic zone that's often defined as the 1% light level. It can be the 0.1% light level, but in any case, that's if you accept the 1% light level, simply 2.8 times the Secchi disk depth gives you the euphotic zone depth, and you can then scale your measurements accordingly. And I would encourage you, if you're not already making regular systematic measurements, they are inexpensive and readily made, and we continue to make them in the California current system. This is an example from a paper that my colleague Madi Karu and another colleague and I published just about a month ago in Limnology and Oceanography. And this draws on a combination of Secchi disk measurements that go back to 1949 in our region, thanks to the CalCoffee program, California Cooperative Oceanic Fisheries Investigations, a fisheries oceanography program that was created. But obviously in 1949, there were not satellites that would give us broad area spatial coverage that would let us infer the optical properties of the water column over a large region. However, there have been many years in the last, say, 30 years when there were both in situ Secchi disk measurements, and this is the graph on the right, the lower axis in situ Secchi disk, and then the y-axis is the satellite inferred Secchi disk measurement. It's I don't need to go into the details, but it's taking the optical attenuation as measured by the satellite and converting that to an equivalent Secchi disk depth. And you can see that over a broad range, the two are fairly highly correlated, and then there's a departure. There's a nonlinear calibration relationship where at very high Secchi disk depths, 30 meters or more, the satellite measurement slightly underestimates that. But so that the two can be combined, the satellite inferred Secchi disk measurement and the actual measurements, Secchi disk measurements, which are made only 4 times per year when a ship is out and they're made at specific locations, whereas the satellite measurements, if it's cloud-free, can cover a very broad spatial area. So the lower graph shows the primary result, which is the changes in optical transparency in this part of the ocean from the early decades, 1949 to 1997, minus the more recent decades, 1998 to 2021. And the shades of blue indicate that the Secchi disk depth has decreased in the last 60 to 70 years, and the shades of pink and red indicate that it has increased. And you can see that over a broad part of the California coast, actually California up into almost to Oregon and then to Baja California in Mexico, the Secchi disk depth has decreased. In other words, that the water column has become somewhat more opaque and less transparent over this multi-decadal time period. And in contrast, in the offshore region, there are many fewer pixels, many fewer measurements, but there's a suggestion that the offshore region the Secchi disk depth seems to have increased, and the water column has become slightly more transparent. And there's some interesting reasons why we think this is the case, but suffice it to say, this is a fairly straightforward measurement to make, that if it's done carefully and systematically, can have important signals in changes in the ocean. And as I said, this has implications for fish feeding success, for phytoplankton concentrations, for alterations in the euphotic zone depth, etc. Okay, so my part 2 of my message is that I think it would be very useful to include a systematic measurement program of Secchi disk depth. I'm going to move right on to the next theme in the interest of time, and this is on a different topic still, and this is the importance of sampling the neustonic layer, the neuston, or the sea air interface layer for suspended microplastics. I mean, I know that there was a very substantial and ambitious program for collecting debris on beaches, nurdles and other plastic debris following the Express Pearl disaster. This is a method to regularly, systematically sample suspended microplastics in the ocean before they arrive on the seashore. I want to point out that many types of microplastics are initially buoyant. They're not all types, but many are initially buoyant and therefore collectible at the sea surface. I say initially buoyant because after they're colonized by bacteria and they're ingested by zooplankton, they can become denser and some of them sink. But specialized sampling devices are required to sample this special sea air interface layer, or the neustonic layer. Conventional plankton nets are not very good for that purpose because they filter so much subsurface water, where what you want to do is just sample that interfacial layer. One of the suitable sampling devices is called the manta net. It's designed to be towed with half the net above the water and half below the water surface, and to have a flow meter, so you quantify the amount of water flowing through the net. And it's quite suitable for collecting microplastics, suspended microplastics. And microplastics are usually defined as net collectible, and nets often have a mesh size of 0.33 millimeters, sometimes 0.55 millimeters, but let's say 0.33 millimeters, and then less than 5 millimeters. So about 1/3 to 5 millimeter material that's suspended at the ocean surface is quite amenable to this type of sampling method. And the manta net was developed here at Scripps actually a few decades ago, developed by someone named Lana Cheng, who was quite interested in pelagic insects called halobates. They're little sea skimmers, and developed this net so that it would skim the surface of the water with the wings. It has buoyant wings, shown in the lower right. The wings have yellow. The yellow wings have foam inside them that keep the net right at the sea surface, and it towed in a quantitative manner for a standardized length of time at a standardized speed. It's possible to get rather good samples for comparison. Now, many people use this for characterizing the neustonic fauna, the specialized organisms that live at the sea-air interface, and not only the marine insects, but also a number of organisms like Velella, they're neustonic snails and other creatures, some Hyperiid amphipods. But then these same samples can be used to characterize plastics. And I thought I would just show you a brief video. I think we're okay on time. This is a deployment. I think I'm going to lower the volume. So this is the manta net. There's the net going over itself over the side. Amy is just reading the flow meter. So you read the flow meter value before deployment, obviously, then after deployment. And because they're at sea, there's a lot of standing around. This is going to take a minute or so before they actually get to the point of they get permission to deploy it over the side. But you can see the yellow wings there, and you see the geometry of the net. It's designed with a bridle that's asymmetrical, so that there's one tow wire that is much longer than the other, and this is intended to tow the net frame out away from the ship so as to minimize the interference of the ship's wake with the sampling. So you obviously, when you're sampling the Neuston, you want to sample clean water that has not been influenced by the motion of the ship. And that's one element of the design. Okay, so I think they've have permission to deploy. It's relatively light. You see the flow meter right in the mouth of the net. It's over the side. And there's actually someone off to the right who's holding lines, who pulls the net to make sure that it lands face down on the sea surface. And we'll just see it. Okay, and then as it's towed, it should move further and further away from the hull of the vessel, so it's sampling basically uncontaminated waters. Okay, so and then this is what it looks like under tow. This is a single frame taken underwater. Part of the mouth of the manta net is above the water's surface, part of it's below, and it's weighted so as to pull it downward against the buoyant action of the wings. And then I had, this is now the recovery. And one of the first thing that's done is that, again, Amy, the technician, comes over and reads the flow meter again and recovers the net on board. Okay, so when these kinds of samples are taken, on the left is an example of a Neuston net sample. preserved material that was collected in the central North Pacific subtropical gyre. And the material on the bottom of the jar is the suspended zooplankton and marine snow, but the material that's floating right up at the surface is largely microplastics, buoyant microplastics. And this was on the edge of the gyre, a very, very long distance away from land. The vial on the right is the sorted material. This is from just a single manta sample taken in the middle of the ocean, far from land. Well, not quite the middle, but very far from land. And you see these a wide variety of different types of abraded particles, looks like little mesh, different colors of plastic debris. And these are the microplastics that have drawn so much attention. And this sampling device is really very well suited to this purpose. Okay, I wanted to briefly mention a method for separating microplastics collected by this method and other methods from non-plastic polymers. But maybe I should just pause for a moment. Are there any questions about manta nets and Newstone sampling in general?
Questions? Do some of the divisions already have these? Yeah, has it been deployed? Yes.
Lahini, you'll have to repeat the question again, please.
I know, I was just wondering if anyone had experience with these types of samples, and they said yes.
Okay.
So, yeah.
Very good. Okay, well then let me move on directly to, I believe that both Chris Reddy and Lahini will be speaking about, or perhaps have already started talking about, some of the analytical chemistry methods that are used for analyzing microplastics. I know that you've done FTIR and I think Raman spectroscopy, there's GCMS and other techniques. An alternative, a much simpler approach that has the advantage that it can be done on large numbers of particles. One problem with FTIR is that each particle that's analyzed has to be prepared, and it can be a little on the expensive side to do very large numbers of analyses. But whereas with epifluorescence microscopy, it's possible to visually differentiate microplastics from other inorganic material and from organic material. It's particularly useful for very small nanoplastic particles. I wish to stress that this is not meant to replace the analytical chemistry, which is much more definitive, but it can be a means to increase the throughput of samples, the number of samples that can be processed per unit time, especially if you're dealing with very small nanoplanktonic plastics. Okay, and to make clear how epifluorescence microscopy works, because that's the approach here, I wanted to draw a contrast between standard transmitted light microscopy, with which we're all familiar. There's a microscope stage with a specimen on top, here shown in blue. There's a light source. It's a polychromatic light source that shines up through the specimen, and the light is either transmitted through the specimen, reflected off it, transmits up through the objective into the ocular, and seen by the eye or by camera. Okay, so this is standard transmitted light microscopy. Epifluorescence microscopy is rather different in that the light is transmitted down through the objective at specific wavelengths, and then the specific wavelengths of fluorescent light are transmitted back up through the objective into the ocular. And while shutting off the light source from below. So let's look over on the right here. We see, again, a polychromatic light source. And in this case, it's filtered. There's an excitation filter. So it's only allowing blue light to pass. The blue light is reflected by a mirror down through the objective, and it contacts the object. It's the specimen here. And in this case, the object is fluorescing green. And so it emits green light, or light at a green wavelength, and the emission filter is now selected to permit only green light to go through. And and that's the light that is then seen by the eye or by the camera. So you have selective excitation and emission. And thinking back to what we discussed about chlorophyll A, you can imagine that if you had a specimen of phytoplankton, live phytoplankton bearing chlorophyll A, if you excited it with blue light, had a blue excitation filter, and you had a red emission filter, you should see bright red fluorescence. In this case, this happens to be a different color scheme. But this is the principle of epifluorescence microscopy. And this is an example of what someone named Jennifer Brandon, who was in my lab, developed, the technique to try to differentiate different, differentiate micro and nanoplastics from other inorganic matter and from organic matter. The panels on the left are bright field illumination or standard transmitted light images of 3 different samples. And by the way, the little black dots that you see here, dark dots, this is simply the pores on the filter, on the polycarbonate filter. Those are not specimens or objects. That's just the background filter. But you can see that it's rather difficult to discern objects against this filter background if you have only bright field illumination. If you excite with 440 to 490, excite with blue light and measure the emission at green wavelengths, you see selected epifluorescence by certain objects in this field. A different wavelength, this is now into the ultraviolet, exciting in the ultraviolet and measuring blue fluorescence, and exciting in the blue and measuring red fluorescence. And what they found was that a combination of fluorescence at these different wavelengths, plus careful analysis of the borders of the objects, whether to determine whether they are likely biological objects or non-living objects, and the evenness of the fluorescence provided a clear diagnosis, or in most cases, a clear diagnosis for fragments. And that the upper row, the material is a kind of plastic fragment. In the lower row, there's short fibers that are under the bright field illumination, are very difficult to discern, but especially under the ultraviolet excitation, they show up clearly. And then there are some long fibers that are basically invisible under the bright field illumination, but under the red fluorescence and blue excitation show up clearly. So this is a method that may be of interest in processing some of your samples. And some people, there's been a lot of attention in the literature to the microplastics that I already described as being between about 0.3 millimeters and 5 millimeters. And very little attention has been paid to the smaller plastic debris, what some would call the nanoplastics. And again, Jennifer Brandon addressed this question of the relative importance of the nanoplastic particles, the tiny things that most people don't collect. You can't collect them with a manta net because the mesh size is too coarse. Most of these particles will flow right through. They're 5, 10, 20 micrometers, and the mesh size is 333 micrometers. So how important are the plastics relative to the microplastics that most people have paid attention to. And what Brandon et al. Found is that in terms of concentrations by number, the nanoplastics are present at 5 to 7 orders of magnitude higher concentrations than the microplastics. This is by number. However, most of these are very small. So if you make the same comparison by surface area, and that's the graph on the right, the nanoparticles sampled in the open ocean in the green bar and nearshore in the gray bar are generally have substantially less surface area than the microplastics do. In other words, by numerical density, the tiny unsampled nanoplastic particles are overwhelmingly dominant. But in terms of the surface area of the plastic material, the smaller number of larger plastic particles are more important. However, many of these very small particles are just the right size that can be readily ingested by suspension feeding zooplankton, by copepods, salps, doliolids, some microzooplankton, and therefore this is a potential pathway of transfer into the food web. So when thinking about quantitative sampling of the plastic debris in the ocean, not only should one think about the microplastics, but certainly consider the nanoplastics. The sampling for the nanoplastic particles is a little tricky. You have to use metal buckets, not, obviously you can't use plastic Niskin bottles or other such water bottles. You need to extend the buckets from metal cables off the ship. You need to filter the water through glass tubing using no plastics. You have to work under a fume hood to minimize the contamination by airborne fibers, because airborne fibers are now virtually everywhere. And you can then freeze the filters and analyze by epifluorescence microscopy or by other methods. Okay, so the epifluorescence microscopy is an approach that you might want to consider, and it might be especially useful if you are thinking about the nanoplastic particles. I think I'm going to skip over this one and just say a few words about the mesozooplankton, but let me stop, and I wanted to go back here. Are there any questions about the epifluorescence method or sampling for nanoplastic particles?
Mark, how many minutes is your next set of slides?
I probably have about 15 minutes more.
Okay. Why don't maybe you can just continue, and then if we have questions, people who have questions can stay and the rest can go have their tea break. Does that sound okay?
Okay.
Is that okay?
I think that's just fine.
Okay.
Okay, so as I told you, I'm a zooplankton ecologist, and I think it would be just too daunting to try to cover important aspects of sampling methods and analysis methods. Let me just mention that there are a variety of methods for sampling the mesozooplankton by nets, pumps, and water bottles, many in situ optical imaging approaches. There are multi-frequency bioacoustic methods, continuous plankton recorder, and recently the advent of autonomous gliders and floats. So there are many approaches that can be considered. And then the analysis of the samples or the images is a separate step from the sampling. And the analysis could be done, taxonomic enumeration remains the gold standard. I hope you continue to train good taxonomists. However, some people prefer to represent the zooplankton community in terms of size spectra. Some categorize them into functional groups or traits. There are optical imaging methods in the laboratory. There certainly are interesting DNA metabarcoding approaches. eDNA is an interesting approach that requires careful calibrations. And then depending on the question, transcriptomic approaches can be very useful, and then experimental incubations. So this is really to say that there are whole fields in these two areas that I'm not going to take the time to go into, but I am going to stress one point, and that's for any type of sampling that you do, it's important to retain samples for multiple purposes. And in our sampling program, in our long-term ecological research site, our standard method is to split samples and preserve half of the sample in buffered formaldehyde. This is for enumeration either by microscopy or by a digital scanning device called the ZoScan, and half of the sample in non-denatured ethanol for molecular genetics. And then for other kinds of purposes, and depending on the research question, we may take separate samples that, or aliquots of these other samples that are frozen in liquid nitrogen or frozen at minus 80 degrees Celsius for rate determinations, for enzymatic assays, grazing assays, molecular probes of diet, etc. So depending on your research priorities, either a formaldehyde ethanol split or perhaps formaldehyde ethanol with some material frozen provides the greatest flexibility for sampling on different sampling methods, different analysis methods going forward. Okay, so, well, Lahini, we are kind of at a juncture, because I could quickly finish, and I think that's another 10, 15 minutes, or people could take a tea break and come back after tea. What would you recommend?
I think the vote is to take the tea. Mark, what's your timeline? Can I just text you and like, because it could be about 15 minutes. Would that be okay?
That would be fine.
Okay, great. All right. So we'll take a break then. And then I will text you. I mean, I'll email you or text you when we're ready to go.
Okay, I'll stand
And
by.
I'll leave everything here. Thank you. Thanks, Mark. Yeah. Yeah, sorry.
I'm sure we'll say this conversation.
Yeah, no, I'm actually, I was told in my.
But the thing I am. I think that. That's just. And I.
Yeah.
Mark, I don't know if you're there, but we have returned, slowly settling into our seats.
All right. Can you still hear me?
Yep, I can hear you. Yep, just
Okay.
thanks for your patience.
That reminds me that it's been a while since I've lived in a tea drinking country.
That's right.
It's a ritual not to be interfered with.
That's correct. Maybe in about a minute or two, Mark, we should be all settled down.
Just give me the word. Actually, let's go on to this.
Okay, Mark, you can get going.
Okay. Well, thank you for returning. What I wanted to touch on is the vital importance of maintaining sample archives for present use, for potential legal purposes if verifications are needed, and for posterity. As I mentioned, I served as curator of a large zooplankton collection for quite some time. And time after time, there would be new analytical methods that people would devise that could be applied to existing samples. And that questions arose about changes in the ocean, about whether the species invasions had occurred, whether there was pre-existing evidence for certain forms for domoic acid producing diatoms in one case, and the well-maintained sample archive is just of crucial importance for contemporary science and for future science. And this is probably more on the side of logistics, and it applies as much to other kinds of sampling as it does to plankton sampling, but I'm referring to that because that's what I work on. So plankton samples require complete comprehensive labeling completed in the field. And the samples should be archived in a safe location with controlled temperature and preferably controlled humidity if possible, and seismic restraints. We had a very difficult situation where the relative humidity in a sample storage archive increased beyond a threshold. We didn't realize it. It resulted in a mold outbreak and in a several-month project to remediate the mold outbreak. So temperature and humidity can be quite important, especially in a warm and humid climate such as yours. Samples should be curated on a reasonably regular basis to ensure that preservation fluids are replenished and adverse pH changes do not occur. And then very importantly, going hand in hand with the physical samples is the electronic databases that are needed to ensure that sample metadata and also the extracted data from those samples can be readily accessed by others. So let me just elaborate briefly on some of these points. Field tow sheets are important to maintain a permanent record of field notes. In this case, you see flow meter readings, and sometimes it's important for someone to be able to go back, see the original, and understand why an anomalous reading occurred. And some of the original data recorded in the field can just be vitally important to sorting things out at a later point, especially if there are anomalies. Clear and permanent labeling of each sample is quite important. We require both lids on the jars, on the jar lids, fully labeled in a permanent archival marker, and then inside labels. And the inside labels are on waterproof stock that is, it's a kind of paper that has a very high clay content and doesn't degrade in solutions. So comprehensive permanent labeling. And more recently, we've adopted the practice of digital barcoding. We do some genetic barcoding, but that's a different subject. The digital barcoding is a means to electronically keep track of particular specimens for sorted material or bulk samples in the case of samples that have a multiplicity of different types of organisms in them. And it's very, very useful for tracking the whereabouts of the samples and for managing the material. And the barcoding, of course, is closely integrated with the collection databases, plural. And we've only recently begun this. I've tried for many years to initiate a digital barcoding effort, and we finally had the grant funding from the National Science Foundation to accomplish this. A permanent sample archive is very useful or dedicated to the storage and preservation of these samples. I've seen situations where samples are considered of secondary importance. They go under someone's shelf or into a back room, but it's really worthwhile thinking about and designing an appropriate archival facility. And I might say it took us many years to be able to develop what we have at Scripps, and many years, and it took multiple grants, but it's surely worth the effort. If you can, at some point, install compact mobile shelving. These are carriages that move on and hand-driven tracks, and it makes for much more efficient use of storage space. This is just one example of a group of samples, plankton samples from our LTR program that are in an organized manner. And this is part of what's being linked to the digital databases. We also have freezer farms for storage of DNA, tissue extracts, and whole specimens for molecular and biogeochemical analyses. This is also an ongoing task to seek the funding to be able to purchase these and maintain them. But this is what maintains the kinds of material of high quality that can be used for future science. And then there's the matter of databases. And this is something that I could go into at length, and I certainly will not take the time. I know that you will have a whole presentation on databasing. I think it will emphasize more the access to data rather than the access to samples. And in the case of our collection, the pelagic invertebrate collection, we have databases for different purposes. There's a searchable database that lists all of our sample holdings. Here's the link, by the way, to this particular page. You can click on this. Anyone can search on the holdings in our collection, find out the sample locations, dates, fixatives, depth sampled, sampling methods, etc. And if they're interested and it's relevant, to request use of the material for their research purposes. But that's a collection management database. There are two other databases that provide guides, informal guides and images. These are intended, the upper one is intended more for the general public, and the lower one is a specialized image archive because of these exceptional larval developmental sequence of a particular kind of lobster that we have here. And then the last three databases are enumeration data. These are data extracted from the samples. And each of these took quite some time to develop, but I would say you are well advised to put time and thought into the design here, and we would be very happy to share with you code that we have and suggestions, and maybe we can learn from you as well. And so my final comments has to do with the general subject of how to be properly prepared to handle spills and other adverse incidents in the future. And I think you've already had discussions about this issue of baseline measurements. We talked, I think it was in May, about this, but ongoing baseline measurements are the real benchmark against which perturbations to a system can be measured. They require systematic, regular sampling, rigorous methods that are calibrated instruments, and people trained and proficient in analytical methods. And so it requires some ongoing effort in that direction, and we're here to help with all of this. My own opinion is that in regards to a baseline measurement program, that a more limited but fully sustainable sampling program is likely to be more useful than irregular spot sampling. That a scaled down sampling program, but one that can be maintained regularly with consistent methodology and developing the expertise within staff, that this would be probably the most useful direction to proceed. And we're very fortunate here in that there was a pre-existing program, I've already referred to it a few times, called CalCoffee, the California Cooperative Oceanic Fisheries Investigations. This program is a fisheries oceanography program. It began with a very real-world economic problem, the catastrophic decline of the California sardine, the Pacific sardine fishery, and the question whether the sardines declined principally because of overfishing or because of changes in the ocean environment. And that simple question has endured up to this day and has spawned many other research programs that don't pertain to Pacific sardine directly, but to the environment in which they live. And I do want to point out one unusual part about the CalCoffee program is that it's a consortium. It's a consortium of 3 entities: the University of California, obviously an academic institution, those of us at Scripps. It's also together with a federal fisheries laboratory, the National Marine Fisheries Service. So a national level government fisheries agency, and then the California Department of Fish and Wildlife, a state level resource entity. And these are 3 different levels of organization and different science bodies that have different purposes and objectives, sometimes even different vocabularies. But remarkably enough, they've partnered and worked well together for going on 8 decades now. And they provide a useful model for how different entities that may have different goals and different societal constraints may have a common interest in the same part of the ocean and can assemble from that common interest a very powerful research program. Okay, and so with that, I'll close and just remind you that what we covered was phytoplankton biomass assessed by fluorometry, Secchi disk as a measure of optical attenuation, Newstone sampling by the MantaNet, especially relevant to suspended microplastics. The utility of epifluorescence microscopy in certain cases for differentiating nanoplastic and some microplastics from non-plastic material. Touched a little bit on really the need for mesozooplankton sampling and analysis without going into that in any detail. And then the vitally important issue of proper archiving of samples, their long-term curation, and the integration of the samples with digital databases to access the information about the samples and the data that have been extracted from them. And with that, I wish you a productive rest of the week. And I'm sorry I'm not able to be there in person.
Thank you, Mark. So before Mark goes to sleep, I was wondering, do people have questions? And I just want to stress that Mark has had probably some of the most experience working with scientific archives, the types of samples that he was talking about. So he has a lot of experience with that, as well as thinking about how to set up long-term monitoring programs and how to build out basic measurements. So I think that's another area if people are interested or have questions or thinking about how to do something like that. Of course, that conversation is kind of happening right now, but I think a really important part of what we all live in and what we do, and I think there's a place where we yeah, and sure they'll take a few questions now. Mark is never far away from some illustration of the plant. He's got so like tiles at school.
Perhaps it's a good thing that I didn't catch all that.
Hi, Marcus. First of all, thank you very much. And it was a pleasure listening to your lecture. And I would like to, if you send any links to your books and publications related to analysis for phytoplankton, it will be very useful for the NARA divisions to change their techniques, I think, because coming to change, we had about 30 years ago, we had a fluorometer, I remember. And now we have a fluorometer attached to a CTD, I think, with both of which were not used very efficiently. And now you have sent us a new one. Thank you very much for that. And that will be absorbing, getting fluorescence instead of absorbance. So I think a new technique will be developed here. And I'm sure we will be able to look after the instrument carefully and learn the procedures and improve our results and reporting. So thank you very much for selecting it and sending it to us. And definitely, I think we are going to make good use of it. So thank you very much, Mark. Do you want to? Yeah. Okay.
Stop it.
Hi, Mark. I'm John from the Division of Oceanography. Can you hear?
I think maybe can you come closer to the.
Oh,
Both of them. Yeah, no, it's good. Just get close. Yeah.
You will see my stomach. That's
Okay, he's been looking at mine the whole time. It's a new stomach to look at.
Can you hear me now?
Yes.
Great. So, since you are an expert in plankton and microplastics, so I would like to know what are the best methods, according to your experience, to analyze microplastics in plankton, zooplankton. Do you have any good method?
I should explain that the people in my laboratory who worked on microplastics in plankton were my students, and my students very quickly know much more than I do. So they're the real experts. But I would say that in the case of the epifluorescence microscopy method, that was developed so as to be able to measure ingestion of nanoplastic particles by cells. Because the particles were too tiny to be able to pick out and analyze one by FTIR. I think that Lahini is probably better able to answer the question about analytical methods, but from what I've seen, FTIR is very informative. And if possible, if you have enough material, I would say that looks like a preferred method, and it's less costly than some alternatives. I would say that the epifluorescence method, as I already stated, it has the advantage of being able to process large numbers of small particles relatively quickly, but it's not unequivocal. There are some polymers, I think polypropylene, for example, that do not fluoresce, whereas most of the others do. So it's not perfect, but it's very useful. I think what I would recommend would be a combination of careful validation of at least a small number of samples, combined with a more rapid analysis, perhaps by epifluorescence.
Okay, great, thank you. That's really helpful, and I have a few more.
Okay.
You told about nanoplastics. So, what would be the filter size, or what kind of filter can we use to collect or filter microplastics? Because now usually for microplastics we use 0.7 or 1.2 microns. Right? So what would be the best filter to be used to analyze nanoplastics?
Well, my student, Jenny Brandon, used 5.0 micrometer polycarbonate filters. You may wonder, wait, isn't polycarbonate a polymer, and doesn't that, isn't that a complication? But it's non-fluorescent. It's almost non-fluorescent, so it doesn't contribute to the background. The second point is that the problem with glass fiber filters is that they're tortuous pore. They don't have a precision pore size of 0.7. Rather, it's the stacking of multiple fibers that statistically results in a median passage of about 0.7 with a broad tail on either side. So if you want an accurate size cutoff, the polycarbonate filters are a better way to proceed. And I would use polycarbonate. I would say that even probably as important as the pore size is this issue of how to keep from contaminating samples. Because you have to use glass, you have to use metal, you have to avoid the fiber contaminants, and the clothing that we wear, much of the material that our food is packaged in, all of these structures are shedding fibers into the air, and it's very, very difficult to keep the background fiber contamination low. So laminar flow fume hood with careful protection is something you have to take precautions for.
Okay. Thanks, Mark. And can I know about vertical sampling of microplastics? If you have done now for horizontal sampling, we have used a manta net, but for vertical sampling to get the samples from different depths, I have used the rosette water sampler and some of the sampling gears. But what would be the best method according to your experience?
Again, that's not something I regularly do, but it's a very important question, because most people think that the plastics are buoyant, they will all be at the surface. But the one slide I didn't show you was a study that my student conducted of Santa Barbara Basin sediments, and she showed that there has been an exponential increase in the plastic microparticles accumulating on the sea floor. This is 500 or 600 meters down. They're obviously negatively buoyant. They make their way to the seafloor, and that since about 1950, there's been an exponential increase in that sedimentary plastic material that closely parallels the worldwide production of plastics. We surely need to know where between the sea surface and the sea floor these particles are distributed. I think your focus on vertically resolving them is very appropriate. I think what I would do would be to work on a closing bottle mechanism similar to a Niskin bottle, but to fabricate it out of something other than a plastic polymer.
So you think the rosette would be a good option?
No,
I'm sorry.
It's a plastic, right?
Yeah, PVC.
PVC.
Yeah. So, I mean, you're saying other than the rosette bottles, right? The Niskins, which are PVC. You're thinking about a metal contraption, perhaps, Mark? Is that what you're thinking?
That's where I would go. Yes.
Yeah.
Okay.
Because you could have something like rubber stoppers. Well, I don't know whether rubber would interfere. It's complicated. You know how to get away from plastics altogether, but yeah, that's the key. I would go with some metallic bottle that with some non-contaminating closure.
No, Mark, I don't remember the name of it. Do you remember? I think they had those, Pete Williams for his DOC and radiocarbon, these Gerard barrels or something. They had some kind of bottle with it. Uwe, do you know what that was? It was a metal, what were those things?
Oh, well, they looked like beer kegs, actually.
Yeah,
Yeah. Well, that is an approach, but that was done one at a time, so that you would bolt a large barrel on, send it down, close it, bring it back. But if you wanted to sample 6 depths, you had to deploy it 6 separate times.
Yeah. Well, the other way to do it potentially is with some kind of in situ pump that doesn't have a plastic where the pump head, because like these in situ pumps, like the McLean pumps, they hit the filter first because they're drawing down. Oh, do you have some? Oh. Yeah. And there are also, McLean makes these other water collection devices. Yeah, I think they're made out of Teflon though, the bags. But anyway, yeah, because I think the McLean pumps are good because it hits the filter head first. So you could have a stainless steel filter head.
Yeah.
Yeah.
We have the pump, but the problem is the depth. It limits to just 20 meters, 50 meters.
Yeah,
per
so,
station.
so the McLean pumps go down to 6,000 meters. They have a titanium, yeah, they have a titanium battery. It's only the battery that matters, right? Well, this man knows
I
everything.
know the wood, I know the wood version, but
Yeah.
I guess it's similar to, they have something
Yeah.
that you can lower on the wire.
Yeah,
Huh?
yeah, we've used those in on the LTR. But those have plastic filter heads, so you need to get metal. Anyway, so that's, I think that might be a way, because then you can also filter large volumes. You know, because if the plastic concentration is low.
Yeah.
So that's what we do if we want to collect
Just
bacterial cells.
So usually, how much of volume?
Oh, but well, that one you can collect. It has batteries. So the one, yeah, so it's a time integrated. So until the battery runs out, which is often for some of them, it's like a couple of thousand, it's 7 liters a minute is how quickly it pumps. I have one that has a 24-hour battery. The other ones are more like 8 hours or so.
Okay.
Yeah.
One more.
Actually, I just wanted to ask, Mark, did Jenny ever do, I kind of remember her doing fecal pellet analyses for plastics, is that right? Did she ever do that?
She's started that.
Okay.
And but it didn't come to completion.
Okay, yeah,
But other people have.
right, because that's another way kind of to get at what's being ingested, right, by the organisms, is to get fecal pellets of some kind.
Well, yes, but that's a somewhat different question from the distribution of suspended particles. Where in the water column they're
Yeah,
concentrated.
no, I was thinking about his question about zooplankton content of nanoplastics.
Oh, okay.
Yeah, yeah.
Thanks, and one more question. Do you find artificial rubber is an emerging problem in your waters? I mean, the plastics that are generated from tire waste, road waste.
Huge.
So
Yeah.
how do you get it counted? I mean, do you discount it from the other types of plastics from the samples, or because it's huge, right? The number is.
Well, it's
Zihini, perhaps you can answer that. I don't know the answer about
so what we, in Southern California, it's actually not so much the rubber from the tires itself, but it's the powder of the rubber. So I haven't come up with a way to discern them from the particulate side, but they have a really, really specific chemical signature. So we see it all the time when we do time series at Scripps Pier. We see that compound, and it can be linked directly to the walls of the tires, the plastic walls of the tires. So that's the only way I've thought about it, is because we can detect the compound that's related to that. But I think it's very hard to visually tell the difference, because sometimes it's very fine. So it's suspended in the water, yeah.
So do you have any method to filter the runoff in the rainwater,
Yeah.
or what kind of solutions that we can suggest? I mean, in our waters, it's hugely running the surface runoff, and even from a rain, we have a lot of.
Yeah, so
Like a sand filter.
no, so what we found is that after a rain event at Scripps Pier, if we collect water samples and solid phase extract them, the compound from the tire powder is detectable by GCMS or LCMS. So that's how we've done it. I haven't actually ever done it as the individual particles. So we see a huge spike after rain events at the pier, even in our part of the world. for that particular compound. So and there's other people who have been studying that compound, I think in Germany and in Washington, because it photooxidizes, I can't remember, it photooxidizes and it's toxic to fish. They found that like salmon have been impacted by the degradation product of that compound.
Thank you so much, Prof.
That's it. And
Thanks to you.
another question, please.
Okay, yeah.
Thank you for your questions.
Okay, one more. Then I promise I'll let you go to sleep.
And so I would like to ask about a best method to analyze.
I'm sorry, could you speak
It's
into the
Joseph.
computer microphone, please?
Yeah.
It's
Well,
better.
actually, my question about the finding of microplastic in the gut contents of the fish. So what is the best method to follow when we are doing gut content analysis of the fish for microplastic as well as for the plankton? And what kind of method that we can use to, what kind of best preservation method that we can use to preserve those gut content for microplastic analysis? That is my question.
It's a very good question, and my answer has to begin with, I'm not a fish expert. Again, it's not very satisfying, but I know that the Scripps Seaplex expedition, when they're first, which began sampling in the open ocean into the Great Pacific Garbage Patch, as it's called, and the Central North Pacific Gyre, one of their first results was based on fish ingestion. And they collected fish by trawls, they preserved them in buffered formalin and did conventional dissections and visual identification of the particles. I think in the case of ingestion by fish, the problem of identification is a little bit easier because, of course, depending on the fish and the size spectrum of prey, the size of prey can vary. But most of the fish that they analyzed ingested somewhat larger sized plastic microparticles that were fairly easy to identify by microscopy. So they didn't have to use epifluorescence microscopy or an analytical chemistry method. So how large are the particles that you're finding in fish guts?
How big are the particles that you're finding?
If we just preserve the sample, so because we
Haven't, okay. Oh, they have the samples, but they haven't had a chance to look at the size distribution. But yeah, I just delivered a digital light microscope that it's linked to a program, Mark, that can do kind of morphometrics of particles to plastics. And I think something like that could be fairly useful for that too. Yeah.
So
But I think
one comment that I would make is that the Brandon method that relied on epifluorescence microscopy, she stressed the importance of carefully examining each particle and looking for something that looked like it was of biological origin or not. Did it have a familiar shape? Did it have a kind of symmetry? Did it have smooth boundaries? Did it have irregularities that are often seen in living material? Oftentimes the plastic has very sharp edges. It's much more uniform in terms of pigmentation. It's more consistent than living material is. So what she recommended was learning the images and learning the appearance of the living organisms, the living prey items first, so as to be able to clearly distinguish based on shape and edges, the non-living plastic materials. And I think if with a trained eye, I think this can be done very reproducibly, especially if you're dealing with larger particles that are a few millimeters in size rather than a few tens of micrometers in size.
Yeah, I think that's a really good point, Mark. The idea that you have to kind of know how to separate out the natural prey items and having a sense of what that looks like for those particular species of fish. I think the other thing that might have gotten lost is the importance of it being preserved in formalin, right, Mark, as opposed to ethanol. The Cplex did it in buffered formalin, is that right?
That's right.
Yeah.
Oftentimes, ethanol is a more effective solvent for pigments. And so if you're using the color or the consistency of pigmentation to identify something as a plastic particle, my understanding is that you're more likely to leach out some of the pigments if it's fixed in ethanol than fixed in formalin.
Yeah, and I think even for the chemistry, it could impact the FTIR measurements, maybe. I don't know if that's the case or not. Okay,
Thank you.
all right, I think, any other questions? Okay, thanks, Mark. Thank you very much. Thanks for your time. Have a good evening.
Thank you all. Have a productive day. Bye-bye.